Ladies and gentlemen, as we are about to begin, please be prepared. I have a note to make before we begin today's seminar. I would like to inform you that I seek your cooperation in completing this seminar. Please kindly turn off your microphone to avoid the noise. Thank you for kind cooperative and attention. And as additional information, this activity does not only take place via Zoom, but you can also watch online via YouTube Departemen Akuakultur Undip. We will provide an attendance link to get a certificate via the chat box during the Q&A session, so don't leave the event until the end. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Her Excellency, the Dean of the Faculty of Fisheries and Marine Sciences, Universitas Diponegoro, Professor Tri Wirnani Agustini, M.Sc, PhD. Respectable, the Head of Bachelor of Aquaculture Study Program, Dr. Insinyur Desrina, M.Sc. Honorable, the Associate Dean Research Training at the University of Tasmania, Professor Barbara F. Nowak, PhD, and respectable all participants of this seminar. First of all, let's thank the Almighty Allah Subhanahu wa Ta'ala, who has given us some mercies and blessings so that we can attend this agenda without any troubles and obstacles. Secondly, salawat and salam may always be given to our Prophet Muhammad Sallallahu Alaihi Wasallam, who has guided us from the darkness to the lightness. I would like to introduce myself. I'm Seto Windarto, lecturer at the Department of Aquaculture, will be your master ceremony in this morning. Welcome to today's seminar, Management of Parasitic Disease in Australian Mariculture, as the World-Class Professor 2021 activities held by the Department of Aquaculture, Faculty of Fisheries and Marine Sciences, Universitas Diponegoro. We would like to thank everyone here for making time to attend this remarkable seminar. Ladies and gentlemen, on this occasion, allow me to introduce you to today's agenda. First, opening by reciting Basmalah. Second, welcoming speech presentation, followed by the question and answer session and closing. Distinguished guests, ladies and gentlemen, let's open this seminar ceremony by reciting Basmalah. Bismillahirrahmanirrahim. Thank you. Next agenda will be a welcoming speech. Allow us to invite Professor Insinyur Tri Wirnani Agustini, M.Sc, PhD, as the Dean of the Faculty of Fisheries and Marine Sciences, Universitas Diponegoro, to deliver her welcome remark. Please welcome Prof. Tri.
Thank you very much and good morning. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. And I'm on behalf of the Faculty of Fisheries and Marine Science, Diponegoro University. Would like to thanks to first of all to Allah for giving us to meet each other today in good health, and also I would like to thanks to Professor Barbara Nowak, PhD from the research training at the University of Tasmania, who will be giving or deliver the speech for today under the program of World Class University program with the World Class Professor. This is our program or belong to the university program that all of the department should have this kind of program to order to improve our knowledge for the staff and also student, and also today we have many participants from different background, including for academician, student, and also for the bureaucrats. And the topic is very interesting, related to the parasitic disease, management of parasitic disease in Australian Mariculture. That will be beneficial for all of us. We can share the information regarding to the management of parasitic disease, because this is very new and improving every day. And we have to follow the progress and development of the management of parasitic disease, especially for fish. And we will learn about how Australian mariculture manage this kind of parasitic disease. So this time is very good topic, and we will hope to share the information from Prof. Barbara to all of us. So thank you also to the head of department, Dr. Desrina, and to the moderator, Dr. Lestari Widowati, to conduct this program for today till the end of the webinar. So on behalf of the faculty, once again, thank you very much and enjoying. And hopefully everyone can have merits and improve the knowledge by joining this webinar. Thank you very much, and wassalamualaikum warahmatullahi wabarakatuh. And once again, and I would like to thanks also to the committee who have give a good cooperation till this program is conducted very well. Thank you very much, and wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Prof. Tri, for your delightful speech. Well, ladies and gentlemen, now proceed to the seminar agenda. This presentation will be delivered by Professor Barbara F. Nowak, PhD, and also this agenda will be guided by Lestari Laksmi Widowati, S.Pi, M.Pi, as the moderator. A brief introduction from the moderator. She is one of the lecturers in the Departemen of Akuakultur, and she got her bachelor until doctoral program at Diponegoro University. And this one information that she just finished her doctoral program last month. Congratulations, Dr. Riri. And she has published many works in reputable journals and has established many collaborations. So without further ado, we invite all the distinguished speaker and the moderator to come to the screen. Dr. Riri, the screen and time are yours.
Thank you, Maseto, for the time. Good morning, ladies and gentlemen. I have your attention. I will make sure that we will have a presentation from our distinguished keynote speaker, Professor Barbara Nowak. Good morning, Professor Barbara.
Good morning, Kuri, and congratulations on your PhD.
Thank you very much. Thank you very much for making sure that is our voice can be heard clearly on your place.
I can hear everything very clearly. Sometimes I can hear other people speaking as well, so maybe it would be good if everybody else turns off microphones, but in general, I can hear you very, very well, and hopefully you can hear me well as well.
Yeah, we are also hear your voice very well here. We hope with all the time of the webinar, the connection will be good. So, ladies and gentlemen, as mentioned by the moderator, I am Riri Widowati. Let me guide these times for nice presentation for Professor Barbara Nowak. But first of all, I ask to read the curriculum vitae from Professor Barbara Nowak. Ladies and gentlemen, Professor Barbara Nowak, PhD, is a professor in the Institute of Marine and Antarctic Studies at the University of Tasmania, where she leads aquatic animal health research group. Her research interest focuses on various aspects of fish health, such as fish parasitology, fish immunology, and fish pathology, including both wild and farm fish. Prof. Nowak have been working on fish health management and disease control. She has been actively involved in Atlantic salmon and tuna health research and collaborated with aquaculture industry worldwide. She is the world expert on amoebic gill disease. Prof. Nowak is recognized as world expert in aquaculture by PubMed's expert scheme. She has been listed as 129th out of 27,800 fisheries specialists and included in the top of 2% of world scientists in any discipline. Prof. Nowak has mentored more than 40 PhD students and co-authored more than 250 peer-reviewed publications. Prof. Nowak is Associate Editor of Journal of Disease, Fish Disease and PeerG and a member of the editorial board of Acta Ichthyologica at Piscatoria. She has received numerous awards for her research and supervision of PhD students. Professor Nowak has been elected Fellow of Australian Academy of Science in 2020. Thank you. And ladies and gentlemen, we are also, we should be very happy here that we have Professor Barbara Nowak for delivering the presentation here. Without further ado, I would like to welcome the presenter, Professor Nowak, to deliver the presentation about management of parasitic disease in Australian mariculture. All right, the time and the screen is yours, Professor Nowak.
Thank you very much for your kind introduction. It's really great to be here, although I would much prefer to be in person. I have been to Indonesia a number of times, only on holidays, I have to say, but I really love diving in Bali and Komodo, and I hope next time I'll be able to visit you myself. But I guess the times are that we have to communicate by Zoom, and hopefully we can do the best out of it. And that also allows us other ways of communication. So not only we can talk, but you can also put comments on chat and communicate that way as well. I will be talking about management of parasites and mariculture. But first of all, just wanted to add that in addition to being a professor at IMAS University of Tasmania, I'm also honorary professor at RMIT University in Melbourne and honorary professor at Aarhus University in Denmark. So hello from Tasmania. I'm not sure how many of you have been here, but I'm currently speaking from Launceston, which you can see on the map here, so you know more or less where I am. And I work at Institute for Marine and Antarctic Studies, which has 3 centers: ecology, biodiversity, oceans and a cryosphere, and fisheries and aquaculture. And I'm at fisheries and aquaculture. We are physically located at 3 different places. Launceston is just one of them. And we also have 2 places in Hobart, Salamanca and Taruna. And I just wanted to thank Sarjito for inviting me today. And he was my first ever master's student. And I also supervised masters of Rainy Turnbull, and she kindly provided this photograph from 2019 conference where she met with Sarjito. So now let's move to the topic itself, some management of parasitic diseases in mariculture, lessons from Australia. I will be talking about fish species, which most likely you are not that interested in, but a lot of methods and way to manage diseases is common for a different cage culture of different fish species. Imagine a world without fish. Imagine a fish disappeared from the world. Fish are essential for at least 5 out of 17 different UN Sustainable Development Goals. If fish disappeared from the world, we would have to find another source of proteins to replace the 17% at least that they are currently contributing. Any fish-based industry like fisheries and aquaculture wouldn't exist, so there'd be massive loss of jobs. Fish disappearance would also cause collapse of many food chains. Fish are important in the ocean as a carbon sink, so we would have a reduced carbon sink. And fish currently contribute 52% of vertebrate species. So loss of fish would also mean loss of biodiversity. So you can see that fish are really essential for the world. And because of that, aquaculture is also essential, as I show you in the next few slides. As you know, the world population is expanding and food production must rise. And by 2050, we will need to increase food production by 70%. So we need better ways to feed the world, and that partly could be done by increasing fish production. Fish products are currently the most traded food products, and they provide between 17 and 20% of animal proteins used by more than 1.5 billion people. Ocean represents 70% of the planet, so obviously it's a huge resource for us to continue using it. However, traditional wild capture fisheries are collapsing. And as you can see from this graph, while the demand for seafood is rising, aquaculture is replacing the gap between what can be provided by wild fisheries and what the demand is. The most popular type of farming is pen farming, and that's partly because establishing pen farming or cage farming or pontoon farming, whichever way you want to call it, is relatively cheap. You don't have to invest in a lot of infrastructure, and you also don't need to lease land. And in most countries, getting access to marine areas is much cheaper than leasing land. So for the industry, the cost is quite low. But obviously there are some issues with pen farming, including biosecurity. It's quite hard to protect the fish against pathogens which are naturally present in the water, because the water just freely flows through the cage. Water quality will also have an effect on the fish, and we can't always control water quality. We can obviously choose good site, which is the optimum for the species, but if there is an algal bloom or jellyfish bloom, it's very hard to control it, and that can affect fish farmed in cages. Farming in fish cages can also have environmental effects, and they can be quite substantial if it's not well planned and well designed. And finally, there can be interactions with wild fish, which can have an impact on wild fish populations. Farmed fish can have quite a lot of different types of parasites, and the main types of parasites that are present on farmed fish include copypods, often called also sea lice, monogenians or skin and gill flukes, blood flukes, amoebae, scuticociliates, and isopods. In most cases, fish in cages are affected by parasites with simple life cycle, which require only one host, the farmed fish. The exception are blood flukes, which, as I'll show you later, require two hosts. Parasitic diseases have very high cost to aquaculture. There is a really great paper published by Andy Shin and his collaborators on economic impacts of aquatic parasites on global finfish production. And this paper has some examples of the costs of parasitic diseases. For example, sea lice in 2009 has been established as costing aquaculture $480 million. Benedinia seriola, which is a skin fluke on Japanese amberjack, in 2001 costed Japan $201 million. Neobenedinia melani, which is a fluke on a Japanese amberjack, currently is costing Japan around $200 million. And one of the parasites that I work on a lot, Neoparamiba perorans, which causes amoebic gill disease on Atlantic salmon, currently costs Australia about $40 million a year, just the management of the disease with no fish dying. In 2006, losses in Atlantic salmon production in Norway costed $12.5 million. And in 2013, losses in Scotland costed $80 million. So you can see from this slide that parasitic diseases can cost aquaculture a lot of money. As I mentioned in my title, I'm going to talk about mariculture industry in Australia, and I'll use just two different fish species as examples. These are the two species that I mostly work on: Atlantic salmon and southern bluefin tuna. Atlantic salmon is a domesticated species. It's a species which has freshwater early life stages, and so we have to have freshwater hatcheries. And it's introduced to marine environment as a smolt after it undergoes physiological adaptation to life in the ocean. The production takes about 18 to 24 months, and it's farmed in Tasmania. It's the biggest aquaculture species in Australia. Southern bluefin tuna is arranged, which means it's caught wild and then put into cages and arranged in the cages. It's a marine fish species, and it's kept only for about 6 months in South Australia. And it's a very high value production, which mostly is exported to Japan. There are some commonalities for management of parasitic diseases in aquaculture, and some aspects of the management of diseases which is common no matter what parasite you are looking at. One of them is biosecurity, and I already mentioned that that can be very challenging in cage culture. Farm design, husbandry, including maintenance of culture environment, diet, so we can try to develop diets which are better to act against parasites. Biological intervention, and that's particularly common in Northern Hemisphere for farmed Atlantic salmon in Norway and Scotland, where cleaner fish are used to remove sea lice. Physical and mechanical measures using physical and mechanical barriers, and I'll talk about it a little bit more later and show you some examples. Parasitic diseases are also treated either with chemical treatments or non-chemical treatments, including fresh water. While there has been a lot of research on developing vaccines against parasitic diseases, there is currently no commercial vaccine developed against parasitic diseases in fish. One of the most successful management of parasitic diseases in aquaculture is selective breeding. And that has been progressing really well for the fish species where we have closed life cycle and domesticated bird stock. So I will first talk about parasites of Atlantic salmon. In farmed Atlantic salmon in Tasmania, the parasites that we have are Neoparamoeba perorans, which is an amoeba shown on the picture on the top. Trichodyna species, that is a ciliate on the picture here. Ceratotoa banksii, which is an isopod. This is a scanning electron micrograph of the isopod. And Sea Lice Caligus langerosters. So all these species have been reported from Atlantic salmon at some stage, but the only one which has impact on farmed Atlantic salmon is Neoparamoeba perorans. So amoebic gill disease is the only parasitic disease significantly affecting Atlantic salmon farmed in Tasmania. What happens is, as I mentioned before, Atlantic salmon in early life stages are in fresh water, and when the fish is small, Neoparamoeba perorans is amphizoic amoeba, which means that it can live freely in seawater, and it will only infect fish and unsalt fish. Risk factors for infections includes high salinity, high temperature, potentially other environmental factors, as well as fish maturation. When the disease outbreaks first started in the late 80s, there were very high mortalities associated with it. However, these mortalities can be alleviated by freshwater bathing. And freshwater bathing is used commercially by the salmon industry, but it increases production costs. When I started to work in Launceston in 1991, there were only 2 geographical locations which have had amoebic gill disease reported, and both of them had first outbreaks in mid-80s. One was in Washington State in Pacific United States, which was described by Professor Mike Kent. And the one in Tasmania was described by late Professor Barry Monday. So they are really the fathers of amoebic gill disease. Since I started to work on this disease, it became much more common, and there's absolutely no association between me working on it and the disease becoming more common. So the current world distribution looks more like this. So we've had amoebic gill disease reported from Atlantic salmon farmed in Norway, Scotland, Ireland, France, Spain, Canada, United States, Chile, and South Africa. And we also had amoebic gill disease reported from other fish farmed in marine cages, in marine environment, including fish farmed in Chinook salmon farmed in New Zealand, marine fish farmed in South Korea, ayu, which is farmed in Japan, as well as some flatfish like turbot farmed in the Mediterranean and in South Africa. So it affects not only Atlantic salmon, but also other marine fish species farmed in marine cages. As I mentioned before, the main way to manage the disease is freshwater bathing. And traditionally, it was done by using cages with a plastic bag, like with a fill, which was lining the cage, was filled with fresh water, and the fish were moved by a pump, fish pump, from the cage into the freshwater bath. And the water was oxygenated the whole time. And after 3 hours, the liner, which was making the bag, was dropped. So the fish were directly transferred to seawater and then either left in the treatment cage or moved to another cage. Now the industry has developed significantly, and they're using wellboats, which you have a diagram here. So this means that there is a big ship, which the fish are moved from the pen into the ship, and then they are treated by fresh water inside the ship in a well, and then moved back to the couch cage. However, this treatment doesn't completely stop the problem. It just removes amoeba for some time, because as I mentioned before, the amoeba are present in the environment naturally. So even if you remove them from the gills of the fish, they will be still in the marine environment. So the graph here is showing numbers of amoeba before freshwater bath. You can see significant reduction on day 1 and day 3, but by day 5, it increases again. So this freshwater bathing may have to be repeated as often as every 10 days or every 2 weeks. Because of that, they've been work on other ways to manage amoeba gill disease. And also in some countries, fresh water is not easily available. In countries where fresh water is not easily available, bath and hydrogen peroxide has been developed. This type of treatment is used against other external parasites, for example, skin flukes, and it can be quite successful. However, there is a very narrow safety margin between how toxic it is to parasite and to fish, particularly at higher temperature. So if you use hydrogen peroxide at higher temperature, it can kill your fish and not just the parasite. Some interesting constructions have been used in Norway, which are called Snorkel Cages. And this is, you can see on the picture on the bottom, the circle in the center of the cage is has a liner in it, so it contains fresh water. Salmon have to come to the surface to gulp air, and if the total surface of the cage, as you can see on this picture, is covered and they can't access surface otherwise, they will have to access surface through the snorkel and be exposed to fresh water for a short period of time. And that can improve a situation with amoeba gill disease. And additionally, fresh water can also kill sea lice. So in Norway, they are really killing 2 birds with 1 stone, because fresh water will also remove sea lice from the fish. Functional diets, functional feeds have been developed by some companies, and some are published in scientific literature showing their potential to remove the amoeba. Experimental treatments have been shown to be moderately successful, and the cost-benefit analysis suggested that it may not be beneficial enough for the industry to invest in commercial use. So there are 2 treatments which are toxic to amoeba and not toxic to salmon, and most likely could be easily used. And that includes butanol and chloramine T. Chloramine T can be also used for other health issues like bacterial gill disease. And then there is another experimental treatment, which is mucolytics, which means it doesn't kill the amoeba, but it makes the mucus very easy to remove from the gills, and the amoeba get removed with the mucus from the gills. So the mucolytic treatment is L-cysteine and can be applied either in water or in feed. And similarly, butanol has been trialed in feed as well. And obviously, as you know, oral treatments have a big advantage over bath treatments because they are not contaminating the environment. Like there's no issue with what to do with it, so the chemical doesn't enter environment. Secondly, if the fish are still feeding when they are affected, they can be delivered directly to the fish. I mentioned earlier on genetic solutions, and that this selective breeding can offer very good progress and very good improvements. In Tasmania and salmon industry, we have breeding programs since 2006, and it achieved significant disease resistance. In addition to the resistance against M.Pi gill disease, this selective breeding program is also working on temperature tolerance, as we are farming Atlantic salmon in high temperatures in Tasmania, and also improved growth and performance. The breeding program has been so successful that we sometimes have problems to get salmon for our experiments, which is not too resistant to M.Pi gill disease. So it's becoming harder to do experiments in Tasmania. In other countries, like in Europe, there is a progress with selective breeding, which reflects Australian experience, so they can achieve some improvement. However, there is an issue in Europe because there are so many other diseases affecting Atlantic salmon that there is not one focus on M.Pi gill disease. They have also to think about sea lice, pancreatic kidney disease, and other diseases. I also would like to mention that there are very new methods to achieve selective breeding and genetic solutions, which has only recently been investigated and experimented on, and hopefully they will be available for work against parasitic disease, to manage parasitic diseases, and that includes CRISPR. Another question we have to ask ourselves, where the amoebae are when they are not on the fish. So when freshwater removes the amoebae from the fish, where are the amoebae hiding? In water, the concentrations of amoebae are very, very low, but even low concentration of amoebae, like 0.1 amoebae per liter, so 1 amoeba in 10 liters of water, can cause outbreak of M.Pi gill disease. Amoebae have been detected on affected farms and sediments as well, but not in unaffected farms. They have been detected in biofouling on the netting of the cages, and it's been suggested but not confirmed that net cleaning may be releasing amoebae from the fouling and possibly reintroducing them to aquatic environment. Other fish species, wild fish species, we've done a lot of studies and we haven't really found any significant infection on wild fish species. However, cleaner fish, and you can see on the picture here 2 species of cleaner fish used in Europe, so lumpfish and wrasse. These fish species have been shown to be affected by AGD, and experimentally the salmon has been infected from those infected fish as well. So they can be a reservoir of amoebae. So I've mentioned at the beginning that there are other parasites present on the gills on Atlantic salmon in Tasmania, which don't cause problems. And they include the isopod and Trichodyna. In Scotland, for example, the gill issues are many. And in addition to AGD, there's also intracellular bacterial infection called epitheliocystis, salmon gillpox virus, and a Macrosporidian desmozoan lepidoptery. If we now look at which of the species are obligatory, so which of the species of pathogens have to have a host to reproduce, in Tasmania it's only Ceratophora banksii, but in Scotland, Norway, and Ireland, everything except for Neoparamoeba perurans has to have salmon to reproduce, so they can't survive in an environment just by themselves. This slide is showing which of these pathogens are killed by freshwater treatment. In Tasmania, in addition to Neoparamoeba perurans, the isopod and the ciliate can also be removed and killed by freshwater. However, in Scotland, in Scotland, Norway, and Ireland, freshwater baths will only remove the amoeba, while the intracellular bacteria, the virus, as well as the microsporidians are protected by host cells, so they're not affected by freshwater bathing at all. So this just illustrates the issues that you might have with treating even the same disease in different geographical locations where you have other diseases present. So to summarize M.Pi gill disease, we have limited management options despite lots of research. I've been working on M.Pi gill disease since early 1990s, and we have tested a lot of different treatments, a lot of different options, but there are really no new practical options. And while we did a lot of work on trying to develop a vaccine, developing vaccine against an ectoparasite, it's very challenging. In some countries, the gill disease includes other pathogens, as I mentioned before, like the virus, bacteria, as well as microsporidium. And in that case, it's called complex gill disease. And so despite we share the Neoparamyba purulans as the pathogen causing amoebic gill disease, there are other challenges, so it's harder to have collaborative research working on the same condition. So the research limitations is access to pathogen. Some people are using cultured amoebae, which lose virulence in the culture, and some people use amoebae from infected salmon, which are maintained in a research facility, but there may be also some changes in the amoeba while it's happening. And then the challenge conditions are very different between lab and field. People under laboratory conditions are usually using very high concentrations of amoebae in thousands per liter, whereas in the real world, in the environment, most of the time, there are fewer than 1 amoeba per liter. Temperatures are very different between what we experience in Australia and what we use for experiments in Australia and Northern Hemisphere, so it's quite hard to compare the situation. There are quite a lot of unknowns for amoebic gill disease. We don't know how much the host will affect the development of the disease. So, for example, if we have a bigger and healthier smolt, it possibly will cause fewer outbreaks and lower severity of the disease. Gill health of smolt of the fish which is introduced to marine cages can also reduce the impact of AGD. Temperature effects have been investigated not in depth, and in particular, the effect of temperature change, increase or decrease, hasn't been really investigated at all. While we know that Neoparamyba purulans is causing amoebic gill disease, there may be changes in the microbiome of the gill happening at the same time. So in addition to the amoeba being pathogenic, we may have a dysbiosis caused either by presence of amoeba, because the amoeba are feeding on bacteria and mucus, or they may be dysbiosis because of other conditions. And we haven't really understood fully the involvement of dysbiosis in amoebic gill disease. Most of experimental infections are infections of naive fish, so the fish which are exposed first time to the parasite. So we don't fully understand how different changes in the host occur in reinfections when the fish is infected multiple times, or co-infections when the fish is infected by different parasites or pathogens at the same time or consecutively. If we compare the management of AGD to management of sea lice, there is not a lot of scope for biosecurity because the fish are farmed in sea cages, and also not a lot of scope in improving farm design. From the point of view of husbandry, there are some functional diets developed again for improving outcomes of AGD. For sea lice, there are functional diets developed against sea lice, and we also have biological intervention using cleaner fish to clean fish of sea lice, and using physical and mechanical measures. And in addition to physical barriers like liners surrounding the cage or lining the cage, there are also lots of trials in Norway in particular using lasers, high temperature, low temperature, and other potential measures, particularly those which can be easily targeted just on the sea lice. So instead of dropping all the fish into high or low temperature, you can just beam the high or low temperature just on the sea lice. Sea lice is mostly treated with chemical treatments, either oral or bath, whereas AGD is mostly treated with freshwater, so non-chemical treatment. There's no vaccine developed either against AGD or against sea lice infections, despite a lot of research and a lot of money spent, particularly on vaccine development against sea lice. And selective breeding has been reasonably successful against both parasitic conditions. I will now move to the second species I was planning to talk about, which is southern bluefin tuna. As you most likely know, there are 30 species of bluefin tuna. Atlantic bluefin tuna, Pacific bluefin tuna, and southern bluefin tuna, with southern bluefin tuna being the smallest of them. And you can see the geographic ranges on the map. And you can also see from the geographic ranges that they do overlap. So southern bluefin tuna overlaps with both Atlantic bluefin tuna and Pacific bluefin tuna. And that means that the wild fish can share parasites. Parasites which are found on ranched southern bluefin tuna, so at the time when they're in ranching, include blood flukes, and that's Cardicola foresteri and Cardicola orientalis. And this is an adult blood fluke which was isolated from the heart of a fish. Caligus chiastos, which is a sea lice present on the skin. Pseudocycnus appendiculatus and Neurophorus brachypterus, both are copypods present on the gill. Hexostoma tinnae, which is a monogenean or gill fluke present on the gills. And Miamensis avidus, which is a scuticociliate, which is a very interesting story that I'll talk about more later. So the parasites which cause significant issues or have caused significant issues in the past in ranched southern bluefin tuna include Cardicola orientalis, Cardicola foresteri, Caligus chiastos, and Miamensis avidus. Miamensis avidus or Enteronima nigricans, two different scuticociliates, have been implicated in causing swimmer syndrome in tuna. In the 1990s, this syndrome was reasonably common in ranched southern bluefin tuna and was causing $2.3 million annual losses. It occurred only when water temperature was below 18 degrees, so in winter. And interestingly, the ciliates were isolated from the central nervous system, from the fluid which you can, and the brain. So what was detected is that the ciliates, these are both species, Euronima nigricans and Miamensis avidus, are amphizoic, which means that they are happily living in marine environment, in the water or on biofouling. However, when the temperature drops below 18, they are somehow motivated to infect fish, and the entry route is through the nose of the fish, through the nares, and they move up olfactory nerves to the brain. And then they are affecting cerebrospinal fluid from which we can isolate them. So on the top picture, you can see just one individual ciliate, and this is the Miamensis avidus, and the cerebrospinal fluid isolated from the tuna. And here is histological section with multiple ciliates, which you can easily recognize because they are the very large cells with a round nucleus. And you can see nervous tissue, lots of red blood cells and white blood cells, and some of those ciliates are full of red blood cells. So they're obviously feeding on the host tissues. Presence of those ciliates in the brain and cerebrospinal fluid causes erratic swimming behavior, where the fish are coming up to the surface, swimming on the surface, then sinking down, and then coming up again multiple times. And because of the swimming pattern, the condition was called swimmer syndrome. Once the parasite gets to the brain of the fish, you can't treat the fish anymore and it will die. So this disease can cause quite a lot of losses, as you can see from this slide. While it was quite common in the 1990s, at one stage, also in the 1990s, there was a big issue with a storm and potentially also toxic algal bloom, which caused significant losses in tuna. And the tuna ranching moved slightly further offshore to a bit deeper water. And since then, swimmer syndrome has been very sporadic. It may affect sometimes, some years there is none at all, and some years it affects maybe 1 pen. And it's obviously significantly related to the location of ranching and quality of the environment where the pens are. Sea lice, Caligus chiastos, is another parasite which has been or did affect southern bluefin tuna in the past. You can see here tuna underwater is definitely the badly affected eye. And all these white dots around the head of the fish are sea lice. So you can see quite a few of them lined up along the upper jaw, around the eye. And the presence of those sea lice was significantly related with blindness and poor condition of the fish. Sea lice have a simple life cycle, which means that they only need one host, and they have two free living stages, nauplius and copypodid. And they have three parasitic stages: calamus, which is attaches to scales and fins, and then periadult and adult, which are mobile on scales and fins. When we were looking at the tuna, we discovered that only 3 of over 5,400 individual sea lice were of the early life stages of calamus, and all the other sea lice on tuna were adults. So it suggested that there must be some other reservoir of infection, most likely wild fish. A large survey of wild fish, many different species, showed that the reservoir was Degen's leather jacket, shown on the picture here, and also on the bottom here, which had very high loads of parasites of the sea lice, in particular early life stages. So those fish were attracted to tuna pontoons by leftover feed, and when they were coming into a ranching environment, they were sharing sea lice with the tuna. We proposed changes to farming husbandry, in particular feed management, also moving the pens further away from the ocean floor and further away from a rocky reef where the leather jackets are more common. And that resulted in significant reduction in infection rates with sea lice, and sea lice is currently not a problem in tuna industry. The main issue in tuna industry currently is blood fluke. There are 3 species of blood flukes known worldwide. And here we have a map showing a distribution of the tuna. So it's southern bluefin tuna in blue, Pacific bluefin tuna in pink, and northern bluefin tuna in yellow. And you can also see the species of Cardicola, which Cardicola is the blood fluke. And that's in Australia, in South Australia, we have Cardicola forstera and Cardicola orientalis. In Japan, we have Cardicola orientalis, Cardicola opistorchis and forsteri. And in the Atlantic bluefin tuna, northern bluefin tuna is Cardicola forsteri, opistorchis orientalis, and one unknown species which has been found on molecular basis. So you can see that the species of the blood fluke are the same in all bluefin tunas. Blood flukes, Cardicola forsteri and Cardicola orientalis, can cause up to 20% mortalities if left untreated. The adult of Cardicola forsteri, which is shown on the picture here, are present in the heart of the fish, and the adult of Cardicola orientalis in the blood vessels in the gill of the fish. The eggs move with blood through the fish, and they usually lodge themselves in the gills. So you can see here eggs of blood fluke in the gills. And then they hatch into free living stage Miracidium, which has to infect intermediate host. Then it undergoes further changes and development in the intermediate host and forms Cercaria, which is the next free living stage, which infects tuna. So this shows a life cycle. So the tuna is the final host where we have the adult parasite producing offspring. And we had intermediate host, and for a very long time we didn't know what the intermediate host was until a large collaborative effort with Queensland University, Queensland Museum, Sardi, University of Tasmania, of course, and tuna industry shown that it was a small marine polychaete, terebellid polychaete Longicarpus modestus. On one hand, that meant that the industry was disappointed because a small worm and sediment is not something we can easily remove from the environment. And if we could remove the intermediate host, that would break life cycle of the parasite, so we wouldn't have the infection. At the same time, it was obvious that if you move into deeper water, if you move the ranching into deeper water and further away from the sediments where the polychaetes are, it's likely that the infection may not happen because it may be too hard for the parasite at the free living stage of Cercaria to infect tuna. It also significantly advanced marine blood fluke research, and particularly for the intermediate host in tuna. And it's inspired Japanese researchers who found the intermediate host just focusing on Terebelid polychaetes, and they found intermediate host for all the different species of Cardicola in the Pacific bluefin tuna. With Cardicola orientalis, which is currently not as big problem as forsteri, so currently the predominant species is Cardicola forsteri. Cardicola orientalis, as I mentioned before, adults are mostly present in branchial arteries in the gills, was first described from Japan in Pacific bluefin tuna, and then detected southern bluefin tuna in 2012. But in archival samples, we got some histology samples which we analyzed. DNA was present since 2005. This map here shows one of the industry experiments where we had most of the companies are close to Port Lincoln. So what happens is the 1 to 4 year old tuna are migrating south along the west coast of Australia, and then they turn the corner and get into Great Australian Bight, where they are caught and then towed back to Port Lincoln in South Australia, and that's where they are arranged. And as I mentioned before, they are just arranged for a short period of time, about 6 months, and fed frozen and fresh bait fish, sardines, and other bait fish, while then pontoons. And this is the map showing Port Lincoln, and you can see the green area is the main ranching area, and we're comparing some pontoons which were in the ranching area with tuna pontoons which were far away offshore. And you can see that near shore, the distance was about 10 nautical miles further offshore. The depth was twice as much, so between 20 meters inshore and 40 meters offshore. There was stronger winds, and maximum wind gust was also stronger. So it was a bit more challenging to farm fish there. But if you look at the results, the mortalities, while initially because of bad weather conditions, they were quite high offshore, the offshore mortalities declined to 0 and remained 0 from week 3 to week 11 of ranching. Whereas inshore, the mortality started to increase at week 7 and stayed plateau around week 7 and only dropped at week 11. And this increase at week 7 is caused by mortality due to blood fluke infection. So the fish offshore had better performance, higher condition index, lower cumulative mortality, and reduced parasite loads with no blood fluke or sea lice present at all. So the offshore farming here, and it could be a possible option, and this also shows that the location of where the farm is really significant and can affect the success or lack of success in fish farming. I've already mentioned that we have 2 species of blood flukes in southern bluefin tuna. And since we couldn't remove the intermediate host from farming environment, the treatment is Praziquantel, which is a well-known antihelminth drug used even for humans against liver flukes. And it's given to the fish injected in a sardine. So the dose of Praziquantel is injected in sardines, and then the medicated sardines are fed to the fish early on after transfer on a couple of occasions. And that reduces significantly blood fluke infection. We still don't know what the intermediate host of Cardicola orientalis is. We don't know what the habitats of intermediate hosts are. Longicarpus modestus, which is the intermediate host of Cardicola forsteri, has been associated with dead shells, but we don't know if they're only close to dead shells or other places. The treatment with Praziquantel kills adult flukes, but not the eggs, so we don't know how to kill the eggs in the host. We don't know how long the free living stages are viable and how much time they have to find the host. And we're also not sure about the Cardicola orientalis origin in Australia. So if we compare management of parasitic diseases in aquaculture, looking at amoebic gill disease, sea lice in the Northern Hemisphere, and blood fluke in tuna, the main similarities between sea lice and tuna is that we have chemical treatment, both for sea lice and for blood flukes, and there are some oral treatments for sea lice as well, and not only bath treatments. Also, because southern bluefin tuna is arranged, and so we don't have closed life cycle, and we don't have a domesticated bird stock, we can't use selective breeding, which so far is the best management option for other parasitic diseases in aquaculture. You can also see the comparisons between AGD and sea lice management, which I've already covered in the previous slide. So management of parasitic diseases in mariculture is still a challenge, despite a lot of research and a lot of money spent on the research. Currently, the best promise is shown by selective breeding, and there are limited husbandry options, and most of them have been used for quite a long time. There are limited treatments. Sometimes they can kill the parasites, but will also kill fish at the same time. Sometimes they may be working very well, but they may be too expensive for the industry to use, or they may have some effect on the consumer, and because of that, they are not. We can't use them on fish which are destined for human consumption. There is no vaccine for any parasitic diseases in aquaculture, and usually there is no silver bullet, so you can't focus just on one option. You can't just use selective breeding and nothing else. And there is a need for integrated pest management, so you have to have like a whole toolkit. And in addition to selective breeding, also know what treatment to use, how to reduce the potential access of parasites to your farm, how to improve fish health so they have best chance to fight the parasitic infections if they encounter them. So it's important, as I mentioned at the very beginning of my presentation, that we can produce fish because they're a sustainable option for food security, and they're also very healthy to eat, and they're an important part of our diet. Fish health is essential for sustainable fisheries and aquaculture, and fish parasites are important to understand because they cause significant losses in aquaculture, but they can also cause significant animal welfare issues, so the fish health and well-being will be affected by parasitic infections. Despite a lot of research, we have limited management options, so it's important to work more on fish parasites. And with some fish parasites, none of the ones I mentioned today, but there are some other fish parasites that may have potential impacts on human health. This slide shows fish species, which we have been studying in our aquatic animal health research group. So I already talked about Atlantic salmon and southern bluefin tuna. The Atlantic salmon is the top farmed species, and southern bluefin tuna is ranched and wild. We have been also recently working a lot on Chinook salmon in New Zealand, not on parasitic diseases, but other conditions. We've done a lot of work on a rainbow trout, in particular using it as a model for salmonids. We've also worked on yellowtail kingfish, Seriola, in particular on gill flukes, as well as epitheliocystis. We've done some work on barramundi, including development of vaccine against bacterial infection, and orange spotted grouper. And in both barramundi and orange spotted grouper, we worked on epitheliocystis. And in the wild, we also worked on striped trumpeter, including health of striped trumpeter in experimental production. Sand flathead, which is a really popular recreationally fished and commercially fished species in Tasmania. And through Aarhus University in Denmark, I also have a collaboration working on fish in Greenland and Denmark, and we focus on Greenland sculpins. The diseases we work on in my group include amoebic gill disease, which I presented in my talk, and we have a challenge model, which means we can experimentally induce the disease. In the past, we worked on two bacterial diseases, Yersiniosis and marine flexibacteriosis, and had challenge models for both of those. Currently, we work on blood fluke infections, and in the past, we worked on sea lice infections in southern bluefin tuna. So the fish are much too big for having any experimental models. And we've done work on epitheliocystis in many fish species, mostly focusing on identifying the agent causing epitheliocystis. I would like to thank you very much for your attention, but before I do this, I would like to acknowledge my collaborators, and particularly PhD students, master's students, honors students. So this slide shows everybody involved in maybe gill disease research for many years. And also this slide shows people who have been involved in tuna research. So I would like to acknowledge all of them, and there's no space or time to read all their names, but they've been really fantastic, and I wouldn't be able to do what I've done without them. So thank you very much for your attention, and I'm happy to take a few questions.
Thank you very much, Professor Barbara, for amazing and informative presentation. That now we are in a question and answer regarding the time. Maybe we have some question that is very interesting for discuss. We are directly go to the question from our alumni from Departemen Akuakultur that already sent a text from the chat box in our Zoom meeting. Thank you, Professor Barbara, for the interesting material. And I would like to looking more answer for from the two questions. The first one, Indonesia has a variety of superior mariculture commodities that are somewhat different from Australia. And have there been a previous research collaboration between Indonesia and Tasmania related to parasitic disease management special for superior commodities developed in Indonesia? For example, white snapper and grouper, or maybe shrimp and etc. The second question is, if there isn't one, I think Undip, Departemen Akuakultur especially, so it is related to our department. Bu Desrina or Pak Sarjito here is the head of the department, maybe can take a note for this question, can initiate a research collaboration with University of Tasmania related this concern, so that there is a focus on solve the problem faced the mariculture industry who develop specific superior commodities in Indonesia. This question is from Mr. Chochon. Thank you, Mr. Chochon, from the question, and I would like to please Mr. Professor Barbara Nowak for answering this question. Thank you.
Thank you very much for the question. So unfortunately, the answer to the first question is no. We don't have currently any collaboration with Indonesia on fish health. I did many years ago. I have approached ACR, which is the Australian funding body for international collaborations, and they said there was no interest in this type of work, but I haven't talked to them for a very long time. I'm not sure how we could get funding in here, but I would be very interested in talking more if anybody is interested in collaboration.
Thank you, Mr. Barbara. So we are from Departemen Akuakultur. Maybe there is an opportunity to have a collaboration. For example, we are here in a coastal area, and for marine also, marine culture area in a tropical countries. The second response is come from Hosna, from Dr. Hosna, PT Singasana Unagi, Indonesia. I want to ask about anchor worm infection in eels after the treatment. We have been reinfected several times. Any suggestion to prevent reinfection after treatment? We have been trying to quarantine the 6th phase.
So I'm not familiar with exactly the infection you are talking about, but it's a really interesting question. And it's another aspect of developing treatment against parasites that we have to also look at reinfections. If you have a land-based farm, which has a full biosecurity and you can treat the water before it goes onto the farm, then obviously you can eliminate a lot of infections and you don't have to worry about reinfections. But most of farming is done still in open cages or flows through ponds. And that means that reinfections will occur. So the two aspects to look for is, first of all, maybe the treatment is stressing the fish as well. So it may be adversely affecting the immune response of the fish, which would be really bad. Maybe it would be good to use some way of immunostimulating the fish, or consider some husbandry treatment to reduce the access of parasite to the farming area. I can't really give you more advice because I'm not very familiar with the parasite and the disease you are talking about. And also I'm not sure what type of culture system you are using. But I would be happy to discuss it in more detail if I have more detail.
Thank you, Professor Barbara. This is another collaboration maybe that we can have for the for the eels. And then the next question, it is come from Prof Budi Prayitno, one of our professor in aquaculture department. Here, Prof Budi asked about the blood slug and sea lice are parasites that are quite often found. What percentage of the cost must be spent to control these parasites, and how much production loss if the parasites are not controlled?
So from the point of view of southern bluefin tuna ranching, the sea lice is no longer a problem. It did cause blindness and loss of condition in the infected pontoons, but it wasn't wide distributed. On the other hand, the blood flukes, as infections are still occurring, at the peak of infections before the treatment was introduced, the infection contributed to about 12% mortality in the industry. After the treatment was introduced, the mortality dropped to 1%. There is some cost for treatment, but the cost is much lower than the cost of 11% mortality of the industry. And I'm really sorry, I don't have numbers other than what I had in the slides to discuss it any further.
Thank you, Professor Barbara. Prof Budi, there is an answer from Professor Barbara. If you are still have a discussion, maybe after the next question, or maybe by the email for the further discussion about blood slug and sea lice as a parasite. And we are great here that we have a lot of questions. So I have to pick it from the chat box, and I have the question here from Dr. Desrina, as our expertise in fish disease also in our department, in aquaculture department. Thank you for enlightening lecturer, Prof. Barbara Nowak. I am curious about Trichodina infection in the marine fish. From what I know, Trichodina is freshwater is usually associated with high organic matter on unhealthy pond condition. Is it the same in the marine culture? Thank you.
Yes, this is a really good point. Trichodina is a ciliate, and it is associated with high organic matter content in the water, with eutrophication in general, and other environmental conditions which are related to high levels of nutrients. And because of it, the risk may increase on farms, because usually farms have higher level of nutrients than other areas. With regard to Trichodina, it's a genus, and they are many different species. I have a friend in South Africa, Professor Linda Basson, and she spent her whole life working on Trichodina. And she showed that if you have one individual fish, there's a different species on the gills and different on skin. So they are not only species specific for the fish, but they're also location specific on the fish. So you can have them on the gills or on the skin or on both. And there will be different species on the gills than on the skin of the same individual fish. So there's a huge range of species. And most of the time, we often see trichodina or trichodinids in gills of fish in histology. Identifying them to species is very difficult, particularly for seawater species, because the salt crystals make the preservation of the sea lice trichodinids, make the preparation of the trichodinids for identification much harder. So freshwater species are a bit easier to identify. But as I said, it's different species for different fish species. So you rarely will get infection from one fish species to another fish species. And if you, for example, catch wild fish and put them in a tank, and the water conditions, the water quality is not very good, and there's high nutrient level, the trichodina, which could be just a few on the fish to start with, multiply very fast and then can cause disease. In low numbers, they don't cause any problems because they are not attached to the fish. They move on the fish. And they usually just feed on the sort of like things which are associated with fish, but they don't bite into the fish. So they don't cause, most of the time, they don't cause any host response. There is no lesion associated with them. And they are usually, all trichodinids are usually ectoparasites on the outside of the fish, but there are a couple of species which have been found in the internal organs and also in urinary bladder of fish. It's a really fascinating genus, and again, I'm happy to discuss it further, but I'm not the expert on trichodina.
Thank you, Professor Barbara, for the answer. Budesrina, I think it is a nice answer from Professor Barbara. And we continue to the last question on my screen, that we have Mr. Faisal for Universitas Erlangga. This is an awesome presentation, Professor Nowak. I would like to ask about the possibility of secondary infection after parasite infestation. Is larger or lower than normal condition? And I have heard one health concept in aquaculture is to tackle sea lice problem in silly salmon farm. And is this concept in use in Australia salmon farm? This is the question, Prof. Nowak.
Sorry, can you read the last sentence again? Because I just lost the sound.
Okay, the second one, I have heard about one health concept in aquaculture to tackle sea lice problem in silly salmon farms. Is this concept is used in Australian salmon farm?
Okay, so I'll answer the last question first. So in Australia, One Health is a really commonly applied concept, but maybe it's understood differently than in Chile, because what we define as One Health is when we talk about health of animals, humans, environment. So it's not just health of salmon and presence of sea lice. So I'm not sure how One Health applies to sea lice. So I can't really address this question properly, because the definition of One Health, I think it's different in Australia than in Chile. And the first question was about secondary infections. And I don't think there is an answer which is correct for all parasites. But if you have parasites which cause lesions, like skin lesions, for example, or gill lesions, of course, it then breaks the barrier, the first barrier which protects the fish against infections, and it can open the fish to secondary infections. So pathogens which are not good enough, not pathogenic enough to be able to break the barrier by themselves, can enter through broken skin or broken gill after a parasite breaks the gill or skin. But it does not apply to all parasites. For example, I don't think amoeba causes lesions which would open gill to other infections. But definitely crustacean parasites like isopods or copypods, they can cause lesions which will cause secondary infections with bacterial viruses.
Thank you, Prof. Nowak. This is from Eric. The question is from Eric, and now we have our student here. Thank you, Prof. Barbara Nowak, for your insightful presentation for today. Let me introduce myself. I am Nadia Elizabeth Hutapea from student of Diponegoro University Aquaculture Department. I'm interested for your presentation about fish health management, especially about fish health essential for sustainable fisheries, aquaculture, and about sustainable development goals. So as we know that the word concern about the microplastic in Indonesia and Australia and other countries. Is there a relationship between the presence of this microplastic and the presence of the parasite for fish in the sea?
Thank you very much, Nadia, for the questions. It's an excellent question. And to be honest, we don't know enough about microplastics in fish. And I'm not aware of any research which would compare parasitic infections and presence of microplastics to see if there is any association between the presence of the two. Just logically, I assume that microplastics would affect fish health, and if they affect fish health, that means that the fish will be more susceptible to any infections, including parasitic infections. But I have no evidence that this is what happens, and particularly in the wild environment. So we would have to investigate it, but it's a great idea for a project.
Okay, maybe for your next research, Nadia, with the microplastic correlation with the parasite disease on the fish. And we are happy now during the time, Professor Nowak, that we have a warm regards from Bapak Dr. Subandiono, our colleague in aquaculture department here. He sent the message in the chat box that, hello, Miss Barbara Nowak, I am Subandiono, your former student from Undip. Yeah, this is the warm regards for you, Prof. Nowak. This is very nice to see you through this forum. Thank you very much for your sound presentation and knowledge.
Thank you for coming. That's very nice of you to participate.
Yeah, thank you, Pak Bandi, for joining this fruitful collaboration webinar between University of Tasmania and Departemen Akuakultur Diponegoro University. We still have a time for some question. Wait a minute. I'm a little bit confused to have a chat on my screen and as well as on my mobile phone. This is from Dwi Prayetno. Dwi Prayetno. Hello, I am Dwi from Tanjung Pura University. The Tanjung Pura is located in Sumatra Island in the west part of Indonesia, right? And does any change for developing natural compound for natural disinfection to fight the gel pathogen?
Yes, thanks for the question. We have done some work on natural compounds. In particular, there was research on garlic, for example, and some herbs. And unfortunately, none of them worked very well. But recently there was a student who was working on olive leaf extract, and the olive leaf extract seemed to be showing some promise, but we currently have no funding to continue. There is a lot of research on natural compounds coming from some parts of the world. It's not really that common in Australia, but I know that there are companies in Australia which work on essential oils, for example, as disinfectants for humans as well. So eucalyptus oil or other oils from native plants. So I think this is definitely a big area of research, as long as the natural compounds can be extracted cheaply enough, because you have to think about it like you will need a big quantity to treat fish, and it has to be cheap enough so it's not too expensive for the farm. People will be prepared to pay much more for something which they use for their own health than to pay much more for a fish because it's treated with natural compounds. For example, I've tried to work with a seaweed company, which was making some natural compounds which were supposed to improve fish health. But the cost of the seaweed compounds, it's okay for medicinal products because people are prepared to pay so much to buy the seaweed compounds, but it's much too high to make them commercially viable in aquaculture. So even at this stage, there is no point trying.
Yeah, thank you, Prof. Nowak, because we are here in our department. The student is intent to have a research in a herbal for some natural potential to be medicine or maybe to against the pathogen in aquaculture in the fresh water and as well as in the marine water for tropical fish. And
I think it's very interesting, and I wish you all the luck, and I hope you will be able to develop natural products for treatment of pathogens, because it is a really good way to go.
Yeah, it is also environmental friendly point of view to have such a herbal medicine or something like that against the disease for the fish. And then we still have two questions from our student. Thank you for the student who already have a good response in this webinar. The student also write the question in English, so it is no need for me to translate. Let me to read the question from Arneta, a student in Aquaculture Department. First thing, thank you so much for your knowledge today. And I just want to discuss that fisheries and aquaculture contribute significantly to food security and livelihood. Fish provide essential nutrition for the people and from the protein and mineral. And this food security is threatened by the climate change and the increase of world population. So the specific effect of climate change on this parameter will vary widely. And then, what is the question here? The lack of oxygen in warmer waters will possibly lead to the extinction of the aquatic animals. I want to asking you about how your opinion and your suggestion for responding this issue regarding of the fish health.
So yeah, obviously fish need oxygen in the water. Some species need much more oxygen than others. So I'm not sure if the question is about wild fish or farming. If you are looking from the point of view of farming, you can focus on fish species which don't require a lot of oxygen. Many years ago, I was in the Netherlands on study leave, and they worked a lot on African catfish, and this catfish could breathe air, so it could be outside water and just breathe air, and they didn't need much oxygen in the water when they were in the water. I know eels don't need much oxygen in the water, but there are some fish species which will require high levels of oxygen, and then if you don't have enough oxygen and naturally in the water you are using, you'll have to introduce oxygen by either aeration or oxygenation of the water, and that will increase production cost. So that's the answer if it's related to farmed fish. With regard to wild fish, if you have dropping levels of oxygen in water naturally because of the combination of climate change and pollution, maybe, then obviously you may have some species which are sensitive enough that it may lead to extinction of some species at some locations. So it can be a significant issue. Most of the time, the species which are going to be extinct will be replaced by other species which don't require oxygen, but they may not be fish. There may be other animal species. So you will have a big change in communities, and maybe if you have completely anoxic conditions, you may also have a loss of life locally in some places. I hope this is not what you are talking about, and I hope the situation is not that bad, and that we will be able to slow down climate change and make sure that doesn't happen.
Thank you, Professor Barbara. We still have 30 minutes on our nice discussion here. The question is come from many aspects, and we have a enrichment of the material from Prof. Nowak. Here we have Bu Desrina. Are you with me, Bu Desrina? Maybe you will have something to discuss since you are also an expert on the fish disease. Burina, you are still muted, Burina.
Okay, I am unmuted myself now.
Yes, please, Burina.
Good morning, Professor Nowak.
Good morning.
Thank you very much. Thank you very much for giving the very nice lecture, very thoroughly lecture. Well, we share. I have. How to say it? I share your concern about the fish disease and the fish control and the management of the disease in the marine culture, because we have the same problem also in the freshwater. Although I've been working with freshwater and some shrimp in the past, actually I work with the parasites of wild fish, because this is very interesting and it's easy to see. I mean, compared to virus, you don't need much equipment to detect the parasites. So I hope in the future we can invite you in person coming to Universitas Diponegoro, and we can discuss further about the collaboration, because actually the university have some programs for collaboration with the international institution. So there is a hope for the collaboration between us, and we can start. This is, I think, the good starting point for us. And Buriri, is there any more question?
Yes, Burina, we still have one question from our student.
Okay, you can go ahead with that one question, because for me, Professor Nowak already explained very well about the trichodyna, and the trichodyna actually you cannot really classify this as parasites, but it is a parasite. So that's the thing with the trichodyna.
Yeah,
and it's very interesting.
You're right. Yeah. It can be present on fish and not have parasitic effect, but then if there are too many of them, they can have an effect and it becomes a parasite. Very good. Yeah.
Thank you, Budisrina, for welcoming Professor
That's
Nowak
very true.
Undip.
So on behalf of the department, okay. Okay.
Yes, Burina, you can continue.
That's all from me. Thank you.
Thank you, Burina, for your warm speech for Professor Nowak and also for the Aquaculture Department in the next collaboration. And Prof. Nowak, I think we still have one question for student. I hope this is not very long question, so I cannot also Pig, what is the real question from her? Thank you for the interesting material, and let me ask a question. And the first one, how to treat or avoid salt bluefin tuna not to get a sea lice? And then the second one, will it have an impact if humans consume the sea fish affected by sea lice? Thank you.
Thanks. So I'll start with the second question. The species of sea lice that I'm aware of, like from Atlantic salmon or southern bluefin tuna, none of them causes problems for human consumption, and they're also present on the outside of the fish. And you can see them with naked eye. So if you buy fish and it has sea lice, you can remove them before you eat the fish, or you can remove the skin. Or if you cook them, they may just add a bit of crunch, but they shouldn't be a problem. So I don't think sea lice are a problem from human consumption point of view. They're a big problem from fish welfare point of view, because I mentioned they can cause blindness and loss of condition. They are also a big problem from causing point of view of causing mortalities, and particularly in Atlantic salmon. Going back to the treatment of sea lice, in Norway, in Atlantic salmon, there are lots of different types of treatment, including different chemicals being used, freshwater bath, and as I mentioned before, laser treatment, beams of hot, very high temperature to kill the sea lice, and other like magnetic fields, all sorts of ideas that they come up with and test on sea lice. But with southern bluefin tuna, because we found that the sea lice wasn't naturally on the tuna, it was coming with another fish species. That meant that we could manage the sea lice infection or reduce or completely get rid of the infection by stopping the other fish species from coming into ranching areas. The leather jacket, which was bringing the sea lice infection to tuna, was attracted by leftover feed. So if the tuna were fed less and there was no leftover feed, that was resulted in the leather jacket not coming. If the tuna were in deeper water, so the leftover feed was further away from the pontoon, from the bottom of the pontoon, so there was no interaction between leather jackets and tuna. Even if there was a leftover feed, the leather jackets were too far from the tuna to infect them with the sea lice. So we were just lucky in the situation with the tuna, because we didn't have to ever treat them. We just could manage the disease by getting rid of the reservoir of the parasite.
Okay, thank you, Prof. Nowak, that I got the information to save that we have consumed the tuna if they're already infected by the parasite. This is no worries to consume it. And Professor Nowak, let me share this last question. This last question from our colleague in an institution in Batam, in Sumatra Island. It would like to know, in the recent years, there has been a parasitic attack of the Lirnanicus S.Pi species that attack grouper fish, especially in a marine cage in their location in the Batam waters. Could you give a suggestion for this case?
So like with any other parasitic infection, you have to understand epidemiology, so where it's coming from, how the infection occurs in the first place, and then also what kills the parasite, how you can treat it, and if it kills the adult parasite, it's going to kill other stages of the parasite as well. So, for example, with sea lice in Norway, one of the ways of trying to avoid sea lice infections is by knowing that the free living stages are present closer to the surface. A barrier, like just a plastic, is put around the cage in shallow water, So the full water flow is only deeper and not shallower, which means that the very living stages of sea lice can't have contact directly with the fish. And that if you know where the very living stages are of the parasite, that's if you can exclude them from the farming environment, that's a really good way to go. And I'm not really familiar with the infection you are talking about, and also the farming system. So if it's a marine cage, as I mentioned before, it's much harder to exclude parasitic stages, but you can still try to use barriers or move the cages to environment where there's more water flow, or it's further from the shore or deeper. And in general, it should help to reduce parasitic infections.
Thank you, Professor Nowak. Maybe your suggestion in a principle can help Ms. Endang to have a healthy grouper phase in the cage system aquaculture farming. Now, thank you, Professor Nowak, once more and more for the answering question, for the great presentation. I have one slide that can be little bit summarized maybe in our discussion regarding your presentation as well as the question from the participant. I'm sorry if I a little bit not from in a fish disease, so I just make summarize in the general. From my note, I have a highlight that I capture from your presentation and the discussion that parasitic disease affecting the fish health and the result in the highly impact of economic and recorded up to $1 million USD. And thus, how to manage it, it has become very important. As far as the technology and the information of how to avoid or how to face up with the parasitic disease, it is very important to identify the life cycle of the parasite itself, the presence of the parasite, as well as the climate that we have and how the farming system that we use and how we handle our management regarding with our species. In order we have the tropical area here in Indonesia, so there is some opportunity that we would like to make our discussion getting rich and rich. So now I would like to say very well, thank you for Professor Nowak, as well as our participants that I recorded from the list come from many universities in Indonesia and many institutions in Indonesia, not only in aquaculture expertise, but also beyond of aquaculture. Also, thank you for the student, our colleagues and our experts.
Dan kira-kira jamak
Thank you, Dr. Riri. Well, that's a wrap. Thank you for a very interesting presentation, Prof. Barbara. I'm totally sure that we got a lot of new knowledge. For your information, there is 100 participants, both in YouTube and also join in our Zoom meeting. Well, ladies and gentlemen, we finally come to the end of today's agenda. Let us close the seminar by reciting Hamdalah. Alhamdulillah. But before ending this morning's activity, we ask you to be able to turn on the camera for the group photo. And Ms. Rosa maybe can help me to capture this moment. Okay, everyone, please turn on your camera so we can see all of your beautiful and handsome face with a great smile. Okay, Bu Rosa, are you ready? Yes. Okay, in the first page, okay, whoa, we have 11 page here. So yeah, be patient one by one. Okay, in the first page, give a big smile. 1, 2, 3. Okay, for the second page, okay, 1, 2, 3. Okay, go to the next slide. 1, 2, 3. Okay, go to the next slide. 1, 2, 3. Okay. The next slides. 1, 2, 3. Okay. Are you ready, guys? 1, 2, 3. Okay, 3 more left. 1, 2, 3. Okay. 1, 2, 3. Okay. Okay, 1, 2, 3. And the last one. Okay, 1, 2, 3. Whoa. It means that it's really interesting, Prof. Barbara Nowak. You can see that there's a lot of participants that enjoying this seminar. And well, those are the agenda we have presented to you all this morning. We hope you learned something. And as the Master of Ceremony, I thank you for your attention and participation. See you on the next occasion. Wassalamualaikum warahmatullahi wabarakatuh. Have a good day, everyone.
Thank you very much, Prof. Barbara Nowak.
Thank you very much.
Bye-bye.
Thank you,
Barbara Nowak.
Thank you, Dr. Barbara.
Thank you.
Professor.
Hopefully, we can see you again in
Thank you, everybody.
Barbara Nowak.
Barbara is good.
Rina and all participants, thanks.
Terima kasih semuanya.
Terima kasih panitia luar biasa
Barbara
telah mengakomodasi acara ini dengan sangat baik. Bu Riri
Ya,
and Tim,
alhamdulillah.
Bu Kaprodi and Tim, Pak Seto yang telah menghandle tadi di kemacetan luar biasa langsung menyalip di tikungan tadi Pak Seto. Luar biasa.
Oke. Terima kasih Bapak Ibu semuanya.
Izin live.
Iya, saya izin live ya. Terima kasih panitia, Pak Seto, Bu Riri, Pak Jito,